So today, ladies and gentlemen, we're going to be learning how to use a microscope. So before we get started, let's go over some basic information about microscopes. All scopes are going to have an arm and a base, and this is how we're going to carry the microscope. So you pick it up with both hands by the arm and the base. They're very expensive. We want to make sure that we don't drop them and damage them. Microscopes have a piece here called the eyepiece, and eyepieces have lenses in them. And the lens is going to magnify something to be bigger than it normally is. So all of the microscopes that we have in this school have an eyepiece with a lens that can magnify something to be 10 times larger than it normally is. In addition to the lens in the eyepiece, we also have lenses on these objectives that you can see me spinning here. And the objectives have different lenses of different powers. The smallest objective that we have has a lens that magnifies something to be 4 times larger than it normally is. Our next objective has a power of 10, which means it magnifies something to be 10 times larger than it normally is. And our strongest objective on this microscope has a power of 43. So that means it's going to magnify something to be 43 times larger than it normally is. However, we need to keep in mind that we also had a lens here in the eyepiece. So if we're trying to calculate the total power of magnification, then we need to take the power of the eyepiece. In our case, those are 10. And we need to multiply this by the power of the objective. So if we are looking at the smallest objective, which is worth 4, then we would have a total magnification of 40. If we have an eyepiece that has a power of 10 and our objective has a power of 10 also, then we would have a total magnification of 100. So something would be 100 times larger than it normally is. If we have a power in the eyepiece of 10 and we are using an objective that has 43 times the magnification of a normal organism, we would have a total power of 430 total magnification. And so that means that our maximum power of magnification is going to magnify something to be 430 times larger than it's normally going to be. When the microscope is plugged in, you have a light source here, and there's a switch to turn the light source on and off. So when we need to focus on something, we're going to make sure that we have this lowest objective, and it is as far away from the stage as possible. So this is the stage. This is where you're going to put your specimen. We have stage clips, which will hold that slide down and make sure that it doesn't move too fast when you're trying to manipulate it. So once we have our specimen under the microscope, we're going to have our lowest objective set, and we're going to use these 2 knobs. The big knob is your coarse focus, and it's big and gray, and you're going to notice that it'll actually move the objectives up and down. We have another knob, which is this little silver one, and this is the fine focus. And this is going to move around the lens in the eyepiece so that we can get our specimen focused. So in order to focus a microscope, you're going to start with the lowest power. The objective is far away from the stage. You look into the microscope and you use the coarse knob to lower this objective toward the stage until you see detail. And once you have your specimen focused, then you're going to center the specimen in your field of vision that you can see. Once it's centered, you're going to switch your objective to the next highest power, and we're going to look through the eyepiece and use this fine focus, this silver knob, to toggle the specimen back and forth until we're able to see detail there as well. Once you have your detail set on this power, you're going to center your specimen in your field of vision, and then we're going to switch over to the next highest power. And once you're in high power, you want to make sure that you're only using the fine focus knob. Anything else is going to move this objective too quickly for you to be able to focus it. And in certain microscopes, you could also damage the lens in the microscope because this coarse knob can move the entire objective and you might end up breaking the slide. So on high power, make sure that you're only using the fine focus, and we'll toggle that back and forth to be able to see detail in high power and also to see different layers of that specimen. We're going to be looking at several different types of cells today, and all of these cells are going to require us to make a wet mount slide. So a slide is a larger piece of glass or plastic that we're going to use to view an organism. And we also have cover slips, and these are tiny little pieces that we put over the specimen in order to protect the specimen and the microscope from touching one another. So in order to make a wet mount slide, first we're going to place a drop of liquid on the slide, just like so. We want to try not to get any air bubbles in that liquid. The next step is we're going to place the specimen on the slide in the liquid. So I'm going to take a piece of onion and I'm going to pull off a single layer of cells from this onion by just peeling back that layer on the inside. So hopefully you can see that there on my finger. This is just a single layer of onion cells. And I'm going to put this in my liquid and try to make sure it goes in there as flat as possible. All right. Once I have my specimen in the liquid on the slide, I'm going to put my cover slip on, and I'm going to lay this cover slip on at an angle, trying to push out all of the air bubbles that might be under this cover slip. So here you can see my slide. Here's my cover slip. Just going to drop that on and try to put it at an angle so that all of my air bubbles get squeezed out. The next thing I'm going to do is place the slide on the stage right here. Use my slide clips to prevent this from sliding around too quickly. And I'm going to focus this by starting on low power with the objective and the stage as far apart as I can get them. And as I look through this, I'm going to lower the objective toward the stage until I see detail. And once I see detail, I can switch to the next highest power and I can use my fine focus from there to try to get more and more detail all the way up to high power. There's also a little slider here on your microscope that will control how much light you're letting go through the slide, and that you can use to increase the contrast of what you're looking at. So you saw me make an onion slide. That's the first kind of cell I'm going to be looking at. We're also going to be looking at a green plant. And so I'm going to make my wet mount slide by dropping a single drop of liquid on my slide. I'm going to take my green plant and I'm going to pull off a single leaf. And we are using elodea because it's only 2 cell layers thick. So it's really easy to be able to get a good picture on the microscope with something that thin. So we put the elodea leaf in the slide, and I'm going to take a cover slip and place it over my elodea at an angle to try to squeeze out all of the air bubbles. And I'm also going to make a slide so that I can look at my cheek cells. And in order to do this, I'm going to use a different kind of liquid, not just water. So I'm going to use something called methylene blue. Methylene blue is going to dye the cheek cells blue so that they show up better under the microscope. It's really hard to see your cheek cells because they are so small and transparent under a microscope, so you can look right through them and never even see them. So the dye is going to help us be able to see those cheek cells. And in order to get my cheek cells, I'm going to take a toothpick and just swab the inside of my cheek. Now, you don't have to put a lot of pressure here. You do need to put some, but if you hurt yourself doing this, well, you're doing it wrong. So you just lightly scrape the inside of your mouth. And those cells are going to come right off into the toothpick. And we're going to take the side of the toothpick that we swabbed with and just roll it around in this methylene blue. And all of those cells are going to come right off into that methylene blue. And I'm going to take my toothpick and make sure that I throw it away because I don't want to leave something with saliva laying around the classroom. Once I've thrown away my toothpick, I'm going to take a cover slip and I'm going to drop that over my methylene blue at an angle and try to squeeze out all of the air bubbles. And then I can put this slide on the stage and go through my focusing routine to see these cells in low to high power. So I'm going to go focus these microscopes on these cells. I'm going to take a few pictures of them from low to medium to high power, and then we'll talk about these different kinds of cells that you're seeing. Now that we've focused on these different plant and animal cells, let's talk about what we were able to see. So this is our onion on low power, and we can see the matrix of cells. They're made up of these little blocks that look kind of like a brick wall. As we increase our power of magnification to 100 times, we're able to see in more detail, and these cells are going to be clearer. We're able to start seeing the nuclei in these cells. And as we go to 430 times magnification, we're able to see the nucleus very well. You can see the cell wall, the space between these cells, and you can see the cytoplasm, the inner goop that makes up the inside of a cell. And that's pretty much all that you're going to be able to see in an onion cell. So this is our elodea on low power on 40 times magnification. As we increase to more detail at 100 times magnification, you can see that we still have the rectangular shape to these cells. And as we go to 430 times magnification, we can see even more detail. So we have the cell wall, which is the space that you're able to see between these rectangular cells. We have the cytoplasm, which is the inner goop of the cell where all the organelles are going to float around in. And you can see these little green bubbles. And these little green bubbles are chloroplasts that are responsible for photosynthesis. Now, we're really not able to see the nuclei of these cells because there are so many chloroplasts in them. Here you're able to see these chloroplasts moving around in the cytoplasm. This is called cytoplasmic streaming. And chloroplasts do this because they don't want to get too much light that would burn the chlorophyll up in them and they wouldn't be able to do their job anymore. So chloroplasts are able to move around inside of a cell in order to prevent getting too much light. If we look at our cheek cells on low power, they don't look like very much, but each one of these little blue dots is a cheek cell. If we increase the power, you can see a bit more detail. We're starting to see the nuclei in these cells. And if we increase to even more power, you're able to see in pretty good detail the nucleus, the plasma membrane. Animal cells do not have a cell wall, so there's no outer coating to these, which is why they have lots of different shapes and not just one type of shape. We can also see the cytoplasm inside of this cell. Let's think of some post-lab questions. So as we increase the power of magnification on the microscope, you see the specimen get bigger and you're able to see it in more detail. The organelles we were able to see in these organisms. In the onion, we were able to see the cell wall. Inside of the cell wall, we had the plasma membrane. We were able to see the cytoplasm and the nucleus. In the elodea, which is also a plant cell, we are able to see that cell wall, the plasma membrane, which is inside of that cell wall, the cytoplasm, but we can't see the nucleus. It is there, but it's being blocked by all of those little chloroplasts, those green bubbles that are doing photosynthesis. In the animal cell, we do not have a cell wall at all. We do have a nucleus and we have cytoplasm and we have a cell membrane, but again, that cell membrane is not enclosed inside of a cell wall. So why do you think the elodea, a green plant, had chloroplasts, but the onion, which is also part of a plant, did not? Well, think about what chloroplasts do. They take in sunlight and process sunlight into carbohydrates during photosynthesis. So the green part of a plant, the leaves, are going to be exposed to the sun and therefore can do photosynthesis. But the onion leaves, which are specialized leaves, specialized to store starch in the plant, these leaves are not exposed to the sun. They are going to be underground. And since they do not come in contact with sunlight, they're not going to be doing any photosynthesis. So they do not need chloroplasts. Key differences we see between plant and animal cells. The plant cells, notice both of them had a cell wall, okay? Animals do not have a cell wall. In the plant cells, we were able to see chloroplasts with the elodea. In the animal cell, we're never going to see those chloroplasts. There are some similarities between plant and animal cells as well. Like we said, all of them have a nucleus and all of them have cytoplasm and a plasma membrane.